Sometimes called the dark reaction, unfortunately, because it doesn't need light directly, but it needs the products of the light reaction. Whoa. It needs the ATP and the NADPH that both came from the light reaction. All right, remember your other sheet here? This sheet, the NADPHs and the ATPs are those guys, are these guys here. Now, this happens in the stroma of the chloroplast. And all of the enzymes and all of the intermediates and so on are already there. But in a nutshell, what happens is this. There are existing compounds already in the stroma that are, you can sort of say, serve as a sort of a starting material. It's called RUMP. It's a 5-carbon sugar. So like any chemical reaction, we need activation energy. We phosphorylate it using ATP, converted to RUBP, which is another 6-carbon sugar. And now the crucial step, or one of the crucial steps of the Calvin cycle occurs. And that's where carbon dioxide combines with RUBP to make an unstable 6-carbon sugar, which then splits into 2 3-carbon sugars. And that step, this incorporation of CO2 into the Calvin cycle, is sometimes called carbon fixation. So that's why a lot of times you'll hear people talk about carbon being fixed, meaning incorporated into the Calvin cycle. And the enzyme that does that is called RUBP carboxylase, and that's kind of an important enzyme because it's crucial to this step, but it's also important because it leads us into this problem that exists, which is the whole photorespiration issue. So we'll talk about that later probably, I'm sure. Anyway, to finish the Calvin cycle, once CO2 combines with this, we make a 6-carbon sugar that's unstable. It splits into 2 3-carbon sugars called PGA. Each of them get phosphorylated again to make 2 molecules of a sugar called diphosphoglyceric acid. That sugar gets reduced using the hydrogens from the light reaction, and lo and behold, I make a somewhat recognizable molecule, I hope, this 3-carbon sugar, PGAL. Now, PGAL can be used to synthesize any kind of 6-carbon sugar, glucose or whatever, okay? And I need 2 of them for each glucose I make. So what we said in class was, in order to keep this a cycle that keeps going and to utilize carbon atoms that are coming from the atmosphere, we need to kind of not only make our glucose, but regenerate the cycle. And the way we do that is by putting a coefficient of 6 in front of everything. So imagine instead of starting with 1 RUMP, I started with 6. And I incorporated 6 CO2s and then reduced them. I'd make 12 PGALs. If I took 2 of them to make my glucose, that would leave you with 10, and those can be rearranged back into our 6 RUMPs again, and I can keep the cycle going.
How is it rearranged to 6 RUMPs?
Well, that's because there are steps here that I'm leaving out. This is that, yeah. You don't have to worry about that, yeah. But just from a carbon atom point of view, since PGAL is a 3-carbon compound, think about it, you've got 10 times 3 is 30, and 6 times 5 is 30. So you have the carbon atoms to do that. It's just a matter of molecularly rearranging them, yeah.
Okay.
So, what we say is that in order for the Calvin cycle to work, I need 6 CO2s that the plant takes in from the atmosphere. I need 12 molecules of NADPH obtained from the light reaction. And I need 12 plus 6, or 18 ATP, to synthesize 1 glucose. So for every glucose I synthesize, those are my sort of raw materials I need, as well as, of course, these intermediate molecules.
And you get these ATPs
from the light reaction, and you get the NADPH from the light reaction. CO2 is coming from the atmosphere.
Okay.
Plant takes it in, right?
Right.
Okay.
And then photorespiration is when?
Well, now, here's the problem. It's been discovered that the enzyme that fixes CO2 into the Calvin cycle is not as specific as it should be. It not only recognizes CO2 and RUBP, but it can also recognize oxygen and RUBP. So in place of CO2 combining with RUBP, oxygen can combine with RUBP, and that leads us into a series of reactions that have been called photorespiration. And what it results in is the use of the breakdown, if you will, of RUBP, removing it from the Calvin cycle, using ATPs without getting any sugar out, and the lack of incorporation of CO2 into the Calvin cycle. So it basically reduces the efficiency of the Calvin cycle. In order to generate an equivalent number of glucoses, I would have to use more ATPs, I would have to take in additional CO2s above and beyond my 6, and I'd have to somehow regenerate my RUMPs or RUBPs to keep this process going. So photorespiration just has a dramatic effect on the overall efficiency of the Calvin cycle. And it's because oxygen, which originates in the light reaction, is acting as a substrate for this enzyme in place of the correct substrate, which is CO2. So, in certain species of plants, this can be so harmful that the efficiency of photosynthesis can be as low as we said, 1%. Now, in temperate plants, where we don't have bright sunny conditions day after day, photorespiration is kind of an annoyance, but it's not a real problem. In tropical and subtropical plants, where the climate is much different and sunny days are very common, photorespiration can be much more destructive. Don't take a picture of me. You're not going to put that on the internet, are you? You are? I'm going to be in it?
Yeah.
Be famous.
Smile.
I don't want to be in it. Why are you getting out of it?
I don't want to be in it.
I'll be in it.
You'll be in it?
Yeah.
Okay. Well, you're only going to share this with your friends, right?
Yeah.
You're
Yeah.
not going to be like,
But Reba has a lot of friends.
I get on MSN and there it is.
There it is. Mr. Kwasny's bio lecture. Right there. So this is
the C4 pathway. That is the adaptation these plants have to photorespiration.
Respiration.
Well, let's take a break and let me ask Jackie for a minute, and then we'll pick this